Hello everyone, welcome to the session of INI-CET May 2024 Biochemistry discussion. I'm Dr. Rebecca James, your faculty with Marrow. So let us go to the questions. As analyzing the questions, almost every questions were simple, straightforward questions except one or two questions. That is the conclusion that I can get from seeing these questions. Okay. So the first question was a match the following question. Actually, this is arranged in the correct way itself. The glycogen accumulation in liver, renal cells, lactic acidemia associated with hypoglycemia. So this is a very straightforward example of which glycogen storage disorder, that is type 1 glycogen storage disorder, otherwise called as von Gerke's disease. So in this particular von Gerke's disease, we can see that there are certain classical features that you should always remember. So the number one is hypoglycemia. Why there is hypoglycemia in this glycogen storage disorder? We know the defect is in glucose 6 phosphatase. So if glucose 6 phosphatase is defective, we know that glucose is not formed. So there is hypoglycemia. When this hypoglycemia is fasting hypoglycemia, what is the reason? Because fasting hypoglycemia, the reason is glycogenolysis is a metabolic pathway that is taking place when the body is fasting, especially in the early stages of fasting, that is 4 to 16 hours without food. So that is first thing, that is there is early morning or fasting hypoglycemia. Then we should also understand that when there is no glycogenolysis, what is the next source of blood glucose? The next source of blood glucose is non-carb substrates. When non-carb substrates is trying to give the blood glucose, we know that the non-carb substrates is entering into what is called as gluconeogenesis. Okay, so when it is trying to enter into gluconeogenesis, what happens is that it is finally getting to the step called as glucose 6-phosphate. Fine. Now this glucose 6-phosphate has to be converted to glucose. So for that also we need which enzyme? Glucose 6-phosphatase, which is defective here. So in this particular case, the hypoglycemia is very severe. Okay, so that is the first biochemical hallmark. The second hallmark is that because of accumulation of this glucose 6-phosphate, what we can see is that this glucose 6-phosphate can enter into HMP shunt pathway. Now, if it is entering into the HMP shunt pathway, then this will increases the production of purines, and this purines will increase. Then what will happen? The excess purine catabolic product will also be there. And what is the catabolic product of purines? It is uric acid. Okay. So there is the second hallmark, that is hyperuricemia. Then what is the next hallmark? The next hallmark is like because of excess glucose 6-phosphate, this glucose 6-phosphate can enter into glycolytic pathway where it can be converted to pyruvate, and the pyruvate can be converted to lactate. Okay. So when this lactic acid, so as a result what happens is there is lactic acidosis, which is the third hallmark of von Gerke's disease. And then we also know that pyruvate can be converted to acetyl coenzyme A. Then why pyruvate is not entering into acetyl coenzyme A? That reason I will tell you. So basically that I will tell you later. So basically what we know is that there is no source of glucose, neither glycogenolysis nor gluconeogenesis. Then the next metabolic fuel the body is searching for is the ketone bodies. So what is the ketone body source for ketone bodies? Then the adipose tissue metabolism will take place, where we know that the triacylglycerol, which is in the adipose tissue, is coming out. The fatty acid is coming out. It undergo oxidation, and as a result what is seen? We can see acetyl coenzyme A is produced, and this acetyl coenzyme A can either enter into TCA cycle or it can enter into ketone body synthesis. And as a result of this excess gluconeogenesis, what we can see is that one of the very important intermediate is depleted here. What is the intermediate? The oxaloacetate. So there is less amount of oxaloacetate, which means that the acetyl coenzyme is not entering into the TCA cycle much. So it is more or less going towards the ketone body synthesis. So this comes the fourth biochemical hallmark, that is ketosis. So we have seen hypoglycemia, we have seen hyperuricemia, we have seen lactic acidosis, and the fourth I said here is the ketosis. Now I will just tell you one more reason why there is lactic acidosis. There is lactic acidosis because the pyruvate is not able to enter into acetyl coenzyme. Why? The reason is because of this excess fatty acid oxidation, there is already excess accumulation of the product which is called as acetyl coenzyme. So as there is an accumulation of acetyl coenzyme, what will happen? This is going to inhibit. This is going to inhibit the enzyme called as pyruvate dehydrogenase. So the pyruvate is all channeled more towards lactic acidosis. Then there is one more biochemical hallmark, and that is hyperlipidemia. It is also explained with the help of this excess acetyl coenzyme. This excess acetyl coenzyme, we know that it is the building block of something else. What is that? The acetyl coenzyme is the building block of fatty acids, right? So there is excess fatty acids, and the same acetyl coenzyme is the building block for cholesterol. Right? So because of these excess fatty acid synthesis and cholesterol synthesis, this lead to the fifth biochemical hallmark, which is called as hyperlipidemia. So these are the biochemical hallmark of von Gierke's disease. So which are the biochemical hallmark? Hypoglycemia, then lactic acidosis, hyperuricemia, then we have seen ketosis, and finally we have seen hyperlipidemia. So going back to this question, von Gierke's disease or type 1 glycogen storage disorder, all these the clinical features are seen. Also seen that there is renomegaly here. In von Gierke's disease, two visceromegalies, two organs where you can see megaly. The one is the hepatomegaly. Hepatomegaly. Then the second megaly is in the kidneys, renomegaly, very characteristic of von Gierke's disease, but very less chance of splenomegaly, less likely to have splenomegaly in type 1 glycogen storage disorder, which is von Gierke's disease. Okay, now going to the next option, that is glycogen accumulation in the lysosomes. So definitely you all know that it is a lysosomal storage disorder, which is a glycogen storage disorder also, and that is Pompey's disease. And what is the enzyme defect in that? The enzyme defect is acid maltase. And which is the megaly that you can see in Pompey's disease? It is cardiomegaly. Okay, then coming to exercise intolerance. Exercise intolerance is a muscle glycogen storage disorder. So here, so that is in McArdle's disease. In McArdle's disease, what is the enzyme defect? It is muscle glycogen phosphorylase. Muscle glycogen phosphorylase or myophosphorylase, which is the next muscle glycogen storage disorder with the exercise intolerance. That is, you can see one is McArdle's disease and the next one is Tarwe's disease. Tarwe's disease. What is the enzyme defect in Tarwe's disease? Enzyme defect is the erythrocyte and muscle phosphofructokinase. So, muscle or erythrocyte form of phosphofructokinase. That is the defect in Tarwe's disease. So, I have explained why a glycolytic enzyme causes a glycogen storage disorder, etc. I have explained in the main videos. So, I am not going too much into the details of how there is a PFK defect is causing a glycogen storage disorder. Please do see those videos. Now going to accumulation of branched chain structure. So in the same, like this I have explained about the von Gierke's disease. In the same picture I am going to show you about a defect in another enzyme, that is the enzyme which converts limit dextrin to alpha-1,4 glucosyl unit, which is a linear polymer, and that enzyme is called as debranching enzyme. Debranching enzyme. So, if there is a defect in this debranching enzyme, then a abnormal glycogen which has lots of branches will accumulate, and that abnormal glycogen is called as limit dextrin. So, that is why this disorder is also called as limit dextrinosis. Limit dextrinosis. One more name is there, it is also called Forbes disease. So, where is the enzyme defect? It is in the debranching enzyme, and you all know about ABCD, right? Anderson disease is causing defect due to branching enzyme, and Cory's disease a defect is in debranching enzyme. Then remember M4M, that is McCord's disease, the enzyme defect is muscle glycogen phosphorylase. One more you can learn like that, H4H, that is Hertz disease. Hertz disease. Hertz disease is also a glycogen storage disorder, and where is the defect? It is hepatic glycogen phosphorylase. Okay, so that is H4H. So these are some of the glycogen storage disorder. It's a very important topic. It is repeatedly asked for many exams. So in this NEET exam also you can expect questions from glycogen storage disorder. I remember one of the NEET previous year question, like almost 2 or 3 questions came from glycogen storage disorder. So that much important is learning about the glycogen storage disorder. So we will see the next question. Familial hypercholesterolemia results due to defect in which of the following? LDL receptor, mitochondrial HMG coenzyme synthase, PDH defect, BCKD defect. So, straight away we know that the answer is LDL receptor defect. So, this belongs to a type 2 hyperlipoproteinemia. Hyperlipoproteinemia according to the Fredrickson classification. So, type 2 hyperlipoproteinemia, which causes familial hypercholesterolemia, has many reasons. One is the LDL receptor. LDL receptor. Then one more reason is there can be a defect in the apolipoprotein which binds to the LDL receptor, and that is ApoB100 defect. So, all these can causes familial hypercholesterolemia. And this familial hypercholesterolemia is the most common hyperlipoproteinemia, and it is autosomal dominant in nature, and because of LDL receptor defect, the lipoprotein elevated is LDL, whereas which is the lipid that is elevated? The lipid that is elevated is cholesterol level is elevated. And one more thing is like the triacylglycerol. What about triacylglycerol? Is it elevated? No. Triacylglycerol level is normal, whereas cholesterol level is elevated. This is what is called as the familial hypercholesterolemia. And remember, familial hypercholesterolemia, there is increased risk of premature coronary artery disease, and family history of premature coronary artery disease are all there in the case of familial hypercholesterolemia. Okay. Then mitochondrial HMG coenzyme synthase. So, it will not lead to familial hypercholesterolemia. Mitochondrial HMG coenzyme synthase enzyme is for what? It is for ketone body synthesis. Right. So, that will not cause a familial hypercholesterolemia. Then PDH defect. PDH defect is the enzyme that convert pyruvate to acetyl coenzyme. So, if there is a defect here, it will not cause hypercholesterolemia. So, that is one thing, because acetyl coenzyme is the building block for cholesterol, that we know. So, if acetyl coenzyme is not synthesized from pyruvate, then definitely it is not going to cause hypercholesterolemia. Then BCKD. BCKD defect, we know it is causing maple syrup urine disease, not this familial hypercholesterolemia. So, clear cut, the answer here is LDL receptor defect. It's a very simple question. I know you all might have answered it right. Congenital lactic acidosis is seen in patients due to defect in the. So, congenital lactic acidosis is due to a defect in an enzyme called as pyruvate dehydrogenase. Then branched chain keto acid dehydrogenase defect, I have already told you it causes MSUD. not a lactic acidosis. Trans ketolase defect is not actually associated with any metabolic disorder, but we should know that the B1 deficiency, that is in thiamine deficiency, trans ketolase enzyme is affected. Only that thing you need to learn. Then isocitrate dehydrogenase deficiency is never causing a lactic acidosis. So what is this congenital lactic acidosis? It is because the pyruvate is not getting converted to acetyl coenzyme A. Okay. So if pyruvate is not getting converted to acetyl coenzyme A, then that is a defect in PDH. So if there is a defect in PDH, then the pyruvate will all be channeled towards another step, that is called lactate. So pyruvate becomes lactate. So in this particular case, there will be accumulation of lactate which causes lactic acidosis. Then this is a very commonly repeatedly asked question, the Lineweaver-Burke plot. It depicts the enzymatic action in the presence of enzyme inhibitors. So I have told you some simple way to remember this Lineweaver-Burke plot and different inhibition. What I have taught you is like in the case of competitive inhibition, you just see the shape. If the two lines are looking like a V, which means that there is no competition. That is a V, VR1, there is no competition, it's a friendly match or something like that. So this is a V shape itself says that it is a non-competitive inhibition. So the first one you can see it is having the shape of a V. So it is a non-competitive inhibition. Then the next I told you there is a competition. When there is a competition, we know that we are like the swords are placed like this, or you remember X. X means there is competition. So if it is an X shape, that itself says that it is a competitive inhibition. So this is a simple way to remember it. So the answer here is non-competitive and competitive inhibition. And if you remember about the kinetic parameters, what about competitive inhibition? In competitive inhibition, we know that the Km value, what will happen? The Km value is increased, whereas Vmax is a constant. Then what about non-competitive inhibition? In non-competitive inhibition, we know that the Km is a constant, but Vmax is reducing. Okay, now just see the x-intercept and the y-intercept. So we see that this is the x-intercept and this is the y-intercept. So in the case of non-competitive inhibition, we know that the Vmax is reduced. So the new Vmax, that is 1 by Vmax dash, is actually moving away from 0, And what about the Km value? The Km value is a constant. So this is the x-intercept. X-intercept is minus 1 by Km. So it will not change because 1 by Km and 1 by Km dash will be the same. So that is why the shape. Now going to this competitive inhibition, we know that competitive inhibition Km value is increasing. So minus 1 by Km is moving closer towards 0. Whereas we know that the Vmax is a constant. So this is the 1 by Vmax. So 1 by Vmax will remain the same because there is no change in the maximum velocity. So this is very simple for you, very commonly asked for many exams. So please do learn these Lineweaver-Burke plot. So all are true regarding the statement regarding folate deficiency except. So there is no question paper without vitamins. So that is the dictum. So this question paper was also not an exception to this. So folate, especially folate, B12, B1, niacin, every time this is very commonly asked. So this time it was a turn for folate deficiency. You can see that all are true statement regarding folate deficiency except. So increased level of homocysteine and normal methyl malonic acid. So we know that this is concerned with about, we'll just discuss about the two enzymes which require B12. So that will explain this increased level of homocysteine and normal methyl malonic acid. So B12, we know there are two different forms. One is the adenosyl B12, which is usually written as ADO B12, and methyl B12. Okay. So we know that methyl B12, what is the role of methyl B12? It is the conversion of homocysteine to methionine. Now, during this conversion, we know that the donor of the methyl group is N5-methyl THFA. N5-methyl THFA is converted to free THFA. Now, this is given to whom? This methyl group is given to B12 and B12 becomes methyl B12. Okay, now this methyl B12 is the one which is methyl B12 gives the methyl group to homocysteine, so that homocysteine is converted to methionine. So, what happens here? If there is a B12 deficiency, then what will happen? There is a functional deficiency of free tetrahydrofolic acid. So, here what happens is there is a deficiency of folic acid. So, if there is a deficiency of folic acid, we know that B12, this reaction will not take place. Okay, this conversion of homocysteine to methionine will not take place. So, what will accumulate? Homocysteine accumulate. Now, what about if there is a B12 deficiency? If the B12 deficiency is also there, then homocysteine accumulate. Why? For this reaction, B12 and folic acid is common, right? So, homocysteine to methionine conversion will not take place, which means homocysteine level is increased both in folic acid deficiency and in B12 deficiency. Now, what about the ADO-B12? The ADO-B12 is adenosyl B12. So, adenosyl B12 in this, for this reaction, the enzyme is methyl malonyl coenzyme A mutase. This is an enzyme which convert methyl malonic acid to succinyl coenzyme A. Okay. So, if there is a deficiency in this enzyme, what happens is this conversion will not take place. And remember, for this reaction, there is no involvement of folic acid. There is no folic acid involved in here, means that this will be affected only if there is a B12 deficiency. So, if there is a B12 deficiency, what will happen? Homocysteine accumulate, methyl malonic acid accumulate, both will accumulate. Whereas, if it is a folic acid deficiency, what will accumulate? Homocysteine accumulate, but methyl malonic acid is not affected because that is requiring exclusively B12. So, just see the first option, increased level of homocysteine and normal methyl malonic acid is definitely a feature of folate deficiency. So, that is a true statement for folic acid deficiency. Then folate deficiency is commonly seen in children with a hemolytic anemia. No, in the case of folic acid deficiency, it is not hemolytic anemia, it is a macrocytic anemia. Hemolytic anemia is usually seen if there is a defect in the RBC membrane integrity. So, in the case of folic acid deficiency, RBC membrane integrity is not affected. So, there is no hemolytic anemia. Then what is affected? There is a disparity between the growth of the nucleus and the cytoplasm. In the case of folic acid deficiency, there is a defective DNA synthesis. So, nucleus maturation will be less, whereas cytoplasm is normal. So, this is called as a nucleocytoplasmic asynchrony is there, right? So, this nucleocytoplasmic asynchrony will cause macrocytic anemia. In the peripheral smear, you can find macrocytes, whereas in the bone marrow, you can find megaloblast. So, megaloblastic anemia or macrocytic anemia is the result, not a hemolytic anemia. So, that is definitely a false statement. It is important to treat coexisting B12 deficiency before initiating folate therapy. So, this is concerned with about an anemia which is because of folic acid deficiency can be created because of B12 deficiency also. So, if this folic acid deficiency is due to B12 deficiency, every time when we are going to treat the patient, first treat the B12 deficiency. Otherwise, what will happen is if you are giving only folic acid in the case of anemia caused due to B12 deficiency, what will happen is all the folic acid will be trapped because there is a B12 deficiency. As long as there is a B12 deficiency, N5-methyl THFA is not getting converted to THFA. So, even if you are feeding the child or feeding the patient with the folic acid, it is not going to increase the folate stores in the body. Always treat the B12 deficiency before treating the patient with the folic acid. So, that is a dictum that we always have to follow. So, that is again, it's a true statement. So, it is important to treat the coexisting B12 deficiency. Otherwise, folate will be trapped. So, treat the B12 deficiency before initiating the folic acid. That's a true statement. Folate supplementation on diagnosis of pregnancy prevents neural tube defects. It is again a true statement. So, the better version of the sentence is like, the ideal thing is like we should start the folic acid supplementation before conception itself, because that is the best time to start a folic acid treatment. So, if you are planning for a pregnancy itself, we have to take a folic acid supplementation. That is the ideal thing. So, if it is not possible, you should start the folic acid treatment, folic acid supplementation as early as possible. The ideal time, I should say, is that when you are planning for a baby itself, you should start folic acid supplementation. Okay. So, again, that is a very correct statement that you all know. So, all the statements are true except the answer here is the folic deficiency is commonly seen in children with hemolytic anemia. No. Next question is, in different ways the question, this fish technique was asked. This time it is like a one-liner. Previously there was a case scenario given and asked what is the technique used. So, fish is a very commonly, very repeatedly asked area. So, this is again a very simple question. Fish technique helps to identify all except. So, we know that the answer here is point mutation. It can detect gene amplification, it can detect deletions like micro deletions, translocations. So, what are the uses of fish? The fish is used to detect structural abnormalities like the translocations, isochromosome, ring chromosomes. These are the structural abnormalities because we are painting every chromosome with a unique fluorescent label. So, when you see the metaphase spread of chromosomes, we can find out any structural abnormality. Second is, if there is a chromosomal deletions like a 5p deletions or Angelman syndrome, Prader-Willi, all these are micro deletions. Credue-Chat syndrome, that is 5p. So, if there is a micro deletions, we can detect it using fish. Then amplification, HER2 amplification, that also can be identified. For example, is what I said, HER2, okay. So, any amplification can be studied using fish. Then aneuploidy, what is aneuploidy? The numerical abnormalities, the monosomies and the trisomies. So, that can also be studied using fish. And it can also help us to find out the correct location of a newly isolated gene to its chromosomal loci. Which chromosome to which band, which arm it belongs to, we can find out using fish. So, these are the uses of fish. Now, what about the other techniques? So, the another question here was the point mutation. So, which are the techniques to detect the point mutations? One is of course the DNA sequencing. So, the different methods are there for DNA sequencing. Sanger sequencing is there, pyro sequencing is there. So, any technique we can use, that is one method. Then RFLP. What is RFLP? That is restriction fragment length polymorphism. So, that can also detect a point mutation. Provided this restriction enzyme should either create or abolish a restriction site. Please do watch the video for more details about the restriction fragment length polymorphism. So, if I want to explain it here, it takes a lot of time. Single strand conformation polymorphism, because certain polymorphisms will create a problem in the single stranded DNA, which can be identified. Likewise, there is DGGE, that is denaturing gradient gel electrophoresis. Again, another technique to detect the point mutation. Then hybridization, like if there is an oligonucleotide probe which is complementary to a particular segment, then whether it binds to that complementary sequence or not, that is oligonucleotide specific hybridization, again another technique. Then microarray. Microarray is also a technique that we can use to study the point mutation. So these are the technique to study point mutations. So basically the mutation detection techniques are divided into two. One is the technique to detect sequence alterations. So whenever we want to, sequence alteration is what we say as the point mutation, which is the most common point mutation is base substitution. One base is replaced by another base, that is called base substitution. So that is the same thing, that is sequence alterations. Whenever we want to study a sequence alteration, first we have to amplify. For that we need PCR. Okay. Now after the PCR we can do pyrosequencing or Sanger sequencing, or we can do restriction fragment length polymorphism. So this is another classification of methods to detect the sequence alteration, which include the point mutation. Then the next thing is like next thing in the mutation detection technique is, the first thing I said is sequence alteration. The second thing is that if there is any alteration in the length, for example, if there is a what you call as repeat polymorphisms are there, trinucleotide repeats are there, then there is a difference in the length, right? So if there is a difference in the length, then that can be detected using amplicon length analysis. So once we amplify it, find out the length of the amplicon, that is one thing, the amplicon length analysis. Then real-time PCR. Why real-time PCR? We know that a real-time PCR, when the amplification is done, a signal is detected. So if there is a long length of the, like any mutation which causes length polymorphism, then what happens is more signals will be produced during the amplification itself. So which will help us to find out the what you call as the amount of the amplicon or the length. So which means that if there is more bases are involved, for example the length polymorphism. In this case, I told you that trinucleotide repeat, length polymorphism, etc. or trinucleotide repeat mutations are also there. So if such kind of things are there, more signals will be emitted. So more signals emitted means when the amplification is done, at that time itself we can find out the signal strength. So this is another way, that is real-time polymerase chain reaction. Then another technique is multiplex ligation probe amplification, MLPA, which I have described in the main videos. Please do watch the video if you want to know about this multiplex ligation probe amplification. So in short, if it is a sequence alteration and if it is a length alteration, the techniques that we use are different. Another very commonly asked question is the study of gene expression. I've told you a very simple way to remember the study of gene expression. Anything that study the RNA is a study of gene expression. So it is Northern blot. We know Northern blot is a technique to detect the RNA. Then RT-PCR. RT-PCR means reverse transcriptase PCR. So RT-PCR, reverse transcriptase PCR is we know that it is also starting with the RNA. So this RT-PCR can be combined with real-time PCR. That is a different thing. But anyway, RT-PCR, if it is there, it means that it is reverse transcriptase PCR. And reverse transcriptase PCR can amplify the RNA. So it is a technique to detect the gene expression. The next is cDNA. cDNA means what? It is the complementary DNA of an RNA. So if we want to detect an RNA, if we have a microarray of cDNA, we can hybridize it with the mRNAs or the RNAs that is like isolated from the cell. So again, just to remember one thing, whatever that study the RNA is a technique to study the gene expression as well. Which of the following micronutrients are associated with a decreased sexual maturation and hypogonadism? So the answer here is zinc. So we'll just see about the various micronutrients that is given in that question. One was copper. So copper, we know copper is required for the enzyme tyrosinase, right? Tyrosinase is required for what? Melanin synthesis. So the copper deficiency can cause hypopigmentation. The next is Wilson's disease. And what is Wilson's disease? It is a defect in the copper transport. In which organ? It is in the liver. Because of mutation in which gene? It is ATP7B mutation. Then Menkes hair syndrome, otherwise kinky hair syndrome or steely hair syndrome. Different names are there. Which is the gene affected here? Here also a copper transport is affected, but the organ here is intestine and the gene is ATP7A. Okay, so ATP7A. Then apart from that, there is something called as Metnick syndrome. Metnick syndrome is a combination of Wilson's disease and Menkes disease. Why? Because here the mutation is in a gene called as A1PS1 gene, A1PS1 gene. So if this gene is defective, what happens is the transport of two proteins are affected. That is the transport of ATP7B and 7A. So because of the transport of these two proteins are affected, it causes a combination of Wilson's disease and Menkes disease, and that is called as Metnick syndrome. Then coming to the zinc, that was a question asked. If there is a mild chronic, so these are all a defect of a mild chronic deficiency. So if there is a mild chronic deficiency, it affects the taste sensation. That is called as hypogeusia. Hypogeusia. Then growth is stunted and immune functions are impaired. So this is one of the mild chronic deficiency. Then if there is a severe deficiency, then it causes hypogonadism, which was asked in the exam. Dwarfism, stunted growth, same thing, dwarfism and hypopigmented hair. So this can also cause hypopigmentation in the hair. Then another thing that was once in one of the exam, this question was asked, that is ectodermatitis entropathica. That is also a zinc deficiency disorder because zinc absorption is defective here. So that is about dermatitis and diarrhea. Dermatitis is usually seen in the mucosa, in the acral regions, that is around the mouth, in the perineal areas, etc. is the site where we can find the dermatitis. Plus there is alopecia. So these are all seen in ectodermatitis entropathica. Again, it's a zinc absorption defect. Then about zinc toxicity. Zinc toxicity is common in welding, like people who use welders, people who use lots of welding a machine, where we can see they are exposed to zinc fumes. So if zinc fumes, it causes respiratory distress. It is an occupational disease which causes respiratory distress, excess salivation, sweating and headache. So these are features of zinc toxicity. Then another micronutrient given in the question was selenium. What are the disorders associated with the selenium? One is Keshan disease, another is Kashenbeck disease. Keshan disease is an endemic cardiomyopathy. Okay, so it is an endemic cardiomyopathy where you can see the dietary intake is very low, like in the regions of China is what is given in the books. So next is Kashenbeck disease. Kashenbeck disease is also a selenium and an iodine deficiency together. Selenium and iodine deficiency together, and here what is affected is not the heart, it is the joint. So it's a chronic joint disease, and that is called Kashenbeck disease. So Keshan disease and the Kashenbeck disease associated with selenium. So the next question was about heme catabolism, where you can see that which of the following is not a heme breakdown product. So if we just see the very first step of heme catabolism itself, we can see all these products. So we know that hemoglobin, if it is undergoing lysis, then globins are released, and what we have is heme. We know heme is a ferroprotoporphyrin, where if you remember there is a structure like this where we can see 4 pyrrole rings. 4 pyrrole rings. This is first, second, third and fourth. 4 pyrrole rings, and they are joined by methynyl bridges. They are joined by methynyl bridges. Okay. So what happens in heme degradation is the very first step, it is taking place in the reticuloendothelial system within the microsomes. What is microsomes? Microsomes are nothing but the smooth endoplasmic reticulum. And the enzyme that act on this ferroprotoporphyrin. So, this is a porphyrin, and in the center what we can find is iron. In which state? In the ferrous state. Okay. Now, this ferroprotoporphyrin is degrading. The very first step that happens is this is acted upon by an enzyme called as HO. HO is what? Heme oxygenase. Now, when this reaction is happening, the methynyl bridge between two pyrrole rings are broken. Which all pyrrole rings? Between the second and the third. The pyrrole ring between second, the methynyl bridge between second and the third is cut, and as a result, what is released is a tetrapyrrole like structure which is called biliverdin. This is a green pigment, plus the iron is released. The iron is released as ferrous form, not ferric form. But we know that there are enzymes which can convert this ferrous form to ferric form. So, later on it can be converted to Fe3 plus form, because the iron, we know that whenever it is transported, this ferrous form is converted to ferric form, and that is how it is transported or stored everywhere. Ferritin, transferrin, in all these places, if you can see that the iron is there in a ferric form. Now, so this ferrous form in the body can be converted to ferric form, then only it can combine with the apoferritin or apotransferrin. So, if it combines with apoferritin, it becomes ferritin and it is getting stored. If it is combining with apotransferrin, then it is in the transport form. So, whenever whatever happens to this iron, this Fe2 plus is converted to the ferric form. That can happen in the body itself. So, whether it is ferrous or ferric, we know that it is a product only. Then, the next very important product is carbon monoxide. This is considered as the only endogenous source of carbon monoxide. Only endogenous source of carbon monoxide is this reaction. And this is the reaction which is taking place in the reticuloendothelial system within the smooth endoplasmic reticulum. So, all these, you can see these all biliverdin, ferrous and carbon monoxide are all the products of the heme catabolism in the very first step itself, that is the heme oxygenase step. And what happens to biliverdin then? Biliverdin is converted to bilirubin. And that enzyme is biliverdin reductase. Biliverdin reductase. Okay. So, and further we know bilirubin is transported in the blood by albumin. It reaches the liver, it gets conjugated, then it reaches the intestine where it is converted to urobilinogen. So, all these heme catabolism I have described in the video, so I don't want to describe here again and again. So, what we have seen is that carbon monoxide is a product, then ferric iron is a product, biliverdin is a product. Then what is this ALA? This ALA is delta amino levulinic acid. What is the source of this ALA? It is like in the heme synthesis, we know very first substrate, succinyl coenzyme A, combines with the glycine and with the enzyme ALA synthase, which is the rate limiting enzyme of heme synthesis, produces delta aminolevulinate or delta aminolevulinic acid. So, it is never a product of heme catabolism, it is a product of heme synthesis. So, all except is not a product of AL. It is a very simple question. Which of the following is not a neurotransmitter derived from amino acid? So, what about GABA? GABA is derived from glutamate. So, glutamate on decarboxylation produces GABA. Okay. Then dopamine. Dopamine is produced from DOPA. DOPA on decarboxylation produces dopamine. When where is this DOPA produced from? It is from tyrosine, and tyrosine is from phenylalanine, which means these are all derived from amino acid. Then 5-hydroxytryptamine, it is formed from 5-hydroxytryptophan, which in turn is derived from tryptophan. So this is also a decarboxylation product. So we can see that 5-hydroxytryptamine. What is 5-hydroxytryptamine? It is nothing but serotonin. Okay. It is derived from tryptophan. Okay. Then we have the last one, creatine. Creatine is a product of 3 amino acids, that is GAM. What are the GAM? Glycine first combined with arginine to produce guanidinoacetate, and this guanidinoacetate is methylated by S-adenosyl methionine, which is getting converted to S-adenosyl homocysteine, and what is formed is creatine, which means 3 amino acids are involved. Again, it is derived from amino acid. So 3 amino acid, glycine, arginine and methionine, which means all these products are derived from amino acid. But the question is, which is not a neurotransmitter? So which are the neurotransmitters here? The neurotransmitters here are GABA is a neurotransmitter, dopamine is a neurotransmitter. From dopamine we should get 2 more neurotransmitters. Which are they? Epinephrine and norepinephrine. Then 5-hydroxytryptamine, again another neurotransmitter. So all these are neurotransmitter. So which is not a neurotransmitter that is derived from amino acid, that is creatine. What is the role of creatine? It is like creatine is converted to creatine phosphate. Okay. Now this is an energy source for the muscle. That is the role of creatine. So that is not a neurotransmitter, but it is derived from amino acid. Which of the following is G6PD deficiency not associated with? Okay, so G6PD, what is the role of G6PD? It is an enzyme in which pathway? In hexose monophosphate pathway. Now, in the hexose monophosphate pathway, in the oxidative phase, what is produced? NADPH is produced. Okay. Now, what's the role of NADPH? 2 main things are there for NADPH. Number 1 is reductive biosynthesis of what? Biosynthesis of steroids and fatty acid, cholesterol, etc. Then what is the second important function of NADPH? It is free radical scavenging. Free radical scavenging. Okay, now we see the question. If there is a defect in G6PD, which is an enzyme in HMP shunt pathway, there is defect in NADPH synthesis. It's a true statement, right? Equal incidence in male and female. That is a false statement. Why? G6PD deficiency is an X-linked recessive condition where only males are affected or males are more affected, which means equal incidence in males and females is a false statement. Predisposes the patient to oxidative stress. Definitely. Why? Because this free radical scavenging is a function of NADPH, means definitely the patient is exposed to oxidative stress. That is correct. So it is true statement. Present as hemolysis. It presents as hemolysis. Why? This free radical scavenging is very important in maintaining the RBC membrane integrity. So, if there is a deficiency of free radical scavenging, RBC membrane integrity is affected, so it causes hemolysis. Then one more thing that we can see here is this NADPH is required to keep the iron in the ferrous state. So, ferrous iron, I have told you, whenever it is getting converted to ferric. But NADPH enzymes, NADPH requiring enzymes are required to keep the iron in the ferrous state. Now, what will happen in the hemoglobin? I am not saying outside the hemoglobin. Inside the hemoglobin, if the ferrous iron is converted to ferric iron, then it becomes methemoglobin. So, if there is methemoglobinemia, it causes anemia, plus there is bluish discoloration of the skin. It is because the oxygen carrying capacity of the hemoglobin is lost. So there is cyanosis as well. So these are all the features. So which is not associated with is the question, and the answer here is B. Which one of the following is not a post-transcriptional modification? So, we know that post-transcriptional modification happens for the RNAs. So, there are different types of RNAs we know. The major post-transcriptional modification that we study is for the messenger RNA or the mRNA. Now, which are the post-transcriptional modification? One is the 5' capping with what? 7-methyl guanosine. Then what is the next one? Poly-A tailing. Okay. Then there is splicing of exons. Splicing of exons. And splicing of exons and cleaving of what is called as the introns. So splicing of exons and removal of introns. Then there is another one which is called as alternate RNA splicing. Okay, so these are all the post-transcriptional modification, plus there is methylation. It is again a post-transcriptional modifications. So all these are post-transcriptional modification, which means this is a post-transcriptional modification. Then you can see methylation, it is a post-transcriptional modification. What about this endonuclease cleavage? Endonuclease cleavage is what you see in this splicing of exons, because we know that if there are two exons, if it is joined by an intron, this exon has to be spliced. If the exon has to be spliced, this intron, the exon-intron junction has to be cut. Who cuts the exon-intron junction? It is by an RNA which is called snRNA, which combines with some proteins and it becomes snRNP. So this snRNP is the one which cuts this exon-intron junction. So it is a cleavage. Now, if you think about this tRNA, we know the tRNA is actually before the for complete modified form of this tRNA. Actually, it is having a extra region in this 5' region. Then there is some extra region in the 3' region. So these extra regions has to be cut before it becomes the complete mature tRNA. Okay, so in that case also endonuclease cleavage is there. Like if you see the ribosomal RNA modification also, ribosomal RNA, if it is formed, it is a long single RNA like that, which has to be cut into 28S, 28S and 5.8S, 5S. All these has to be cut from the long RNA that we are getting, which means endonuclease cutting is there for ribosomal RNA. Then if you see the post-transcriptional modification of microRNA, dicer DGCR8. Dicer and DGCR8 are something which cuts the microRNAs. So that there also what we see is an endonuclease cleavage. So we can see the endonuclease cleavage invariably in almost all RNA post-transcriptional modifications of the RNA. Then what about glycosylation? Glycosylation is not a post-transcriptional modification, it is a post-translational modification. Translational modification. If the question is instead of endonuclease, if it is a protease, then it is a post-translational modification. If it is a protease cleavage. Protease cleavage is something which happens as a post-translational, that is to the protein. That is why protease. But endonuclease means definitely it is something that happens to the DNA or the RNA. So in this case, RNA is definitely a ribonucleotide. It is a nucleotide. So endonuclease cleavage is for RNA, not for a protein. But glycosylation is a post-translational modification. So the answer here is glycosylation. Now going to the next question, which type of mutation result in reversal to wild type of phenotype when mutant gene is suppressed? Maybe that is the only question that you find a difficult one in that the whole INI-CET question paper. in the biochemistry session I mean. Okay, so deletion of mutated gene, frame shift of mutated gene, addition of another normal gene, mutation in tRNA. So all these are a kind of one or the other kind of mutation. Deletion of a mutated gene is a kind of mutation. In all these things, it will not reverse a mutant gene to a wild type gene. Mutant to a wild type, that is, which will never happen. So what is this? It is actually something called as a suppressor mutation. Suppressor mutation. Okay, what is the suppressor mutation? We have learned about mutation. Mutation is something that happens in the DNA sequence, deoxyribonucleotide sequence, if one base is replaced by another base, or if certain bases are deleted, or if it is amplified. So these are something which happens in the DNA, that is called a mutation. But what is suppressor mutation? Suppressor mutation is DNA mutation is there, but there is something which is suppress this DNA mutation. How this DNA mutation can be suppressed? It is by something else undergoing a mutation which is actually preventing this it is preventing it from producing a phenotype. Genotype is something that happens in the DNA. When it affects the protein and when it affects the external features or externally expressed protein, then it is called as a phenotype. Okay, genotype, phenotype. Now, there is a mutation in genotype, but the phenotype, which is the one that is expressed, is not affected. Why? Because there is something which can suppress this mutation. So, they are called as suppressor mutation. Actually, so suppressor mutation is helping us, which preventing this genotype not to produce a abnormal phenotype but a normal phenotype. Okay, so mutation is there in the DNA, but the phenotype is not affected. So this kind of, or what is this kind of suppressor mutation? This suppressor mutation takes place in the tRNA. tRNA. So what is it? I'll just show you that with the image. Just see this codon. Here it is written CAG. CAG is a codon for glutamine. So definitely the tRNA that is coming and binding with this codon should carry glutamine, right? So if this tRNA has to bind to this glutamine, then it should bind with this codon CAG. So what is there in the anticodon arm of this tRNA? There is, you can see from this side, if you see, you can see G, if we are, I'm seeing it, I mean, if I'm reading it from 5' end, it is CUG. I'm just reading it from the other end, that's CC. So you can see GUC here, okay. This G we know it can combine with C, U can combine with A, and C can combine with the G. We know ATGC, or in the case of this is RNA, so it is AUGC. We know that they are all complementary pairs, so they will bind. So this is what should happen actually. So this is a wild type of genotype, and this is what you can see, the protein that is formed as the phenotype, or what you can see externally as a symptom or clinical feature, whatever normal feature we are seeing, that are all the normal phenotype. What is the expressed character, that is called as the phenotype because of the proteins that are synthesized. Okay, fine. So we can see that in this particular translation glutamine is produced or added. It is because of this codon anticodon interaction. Now, what happens here is, instead of the CAG, you see here, what happens is the cytosine is converted to U. The most common, this is one of the most common mutation, that is called a spontaneous deamination. Spontaneous deamination of cytosine to uracil. So, C to U means that this has become a UAG. What is UAG? U-argon. What is U-argon? It is a stop codon. Okay. So this stop codon will create what is called as a truncated protein. Truncated protein. Okay. And this mutation is called as a nonsense mutation. So this kind of suppressor mutation happens in nonsense mutation, which means a coding codon is converted to a stop codon. So what we expect on the other side is a truncated protein is produced. But what happens here is the tRNA is helping. How the tRNA is helping? In the tRNA, another mutation is happening. So just see what happened in the tRNA. In the tRNA, the G, this G is replaced by A. So it is a helpful mutation. How? This becomes AUC now. So this tRNA, like in this tRNA, G becomes A and the same tRNA is carrying glutamine. At the same time, this UAG will not act as a stop codon because you can see this A can form a pair with U, then likewise U to A and C to G. So there is a base pairing that is happening which makes this UAG not a stop codon but it is a coding codon which is joining with the glutamine. So this kind of what you call as a suppressor mutation. This is otherwise called as a secondary mutation because primary mutation is in the DNA. So the secondary mutation which is called as a suppressor mutation that happens in this tRNA. So the same glutamine will bind. So my question is whether the phenotype will change. No. Whether a truncated protein is produced. No. No truncated protein is produced. So these kind of mutation, a kind of secondary mutation which actually suppress the genotype, this genotypic mutation is not corrected, right? That mutation is still there, but the phenotype it won't be there because there is a secondary mutation or a suppressor mutation that suppress this primary mutation. So these are called as the suppressor mutation. It is a very useful technique that we use for tRNA medicine. So I just introduce you to what is this tRNA medicine. I know this picture is not so clear to you, but still I just explain what is it to you. There are various kind of mutation that can happen in the genotype. So what is given here is a nonsense mutation, or frame shift mutation, or missense mutation. Okay. Now the suppressor mutation that I have just told you is a normal thing that can happen in our body where the suppressor mutation is actually protecting our body from an abnormal protein, a truncated protein. But we are genetically engineering certain tRNAs because if we know that there is a nonsense, this is the first example is there is a nonsense mutation in the genotype. By DNA sequencing, we find that this disease is caused because of a nonsense mutation. Then what we will do is, we will genetically engineer a tRNA so that its anticodon arm can bind to this nonsense codon or a what you call as a stop codon. So that is what is shown here where a genetically engineered tRNA whose anticodon can bind to this mutated region and it codes for the same amino acid so that there is no change in the phenotype. Okay. Now I'll tell you one more example. Frame shift mutation. What do you mean by frame shift mutation? Insertion or deletion of single bases. So if there is insertion or deletion of single bases, what will happen? The whole reading frame is garbled. The example that I have given in my videos is the cat eat rat. Okay. This frame shift mutation means I'm just telling an example of an insertion where before this C, Z is added. So these are the codons. So we know that codons are all 3-letter words, but in the 3-letter word an abnormal letter is added that is called Z. So what will happen? The Z cat eat rat. So it is a frame shift mutation where we can see the first translation will these 3, the next it will read these 3, which means the entire reading frame is garbled. So what we will do is that we will make a genetically engineered tRNA which is having 4 nucleotides in the anticodon arm. Okay, 4 nucleotides in the anticodon arm. So we know that if this codon is suppose if this codon is supposed to code for glutamine, then what we will do is, we are having a genetically engineered tRNA which carry glutamine in it in this acceptor arm. At the same time, this anticodon arm is complementary to the ZCAT or ZCAT, which means this will bind, which means this will suppress this frame shift mutation, because at that position what this cat has to, or what that cat codon has to produce will be there in this tRNA. So this will go and bind here, which means there is no frame shift. Again, this is a medicine, a tRNA medicine that we can use for the treatment. Then missense mutation. Missense mutation can also be corrected using the same tRNA. For example, if a missense means in this example, if I am saying the cat eat rat, okay, these are all, suppose these are all the codons, okay. So in the C, if the C is replaced by Z, then the codon has changed to cat to ZAT, right? So ZAT may be a codon for another amino acid. So that is called as a missense mutation. If the ZAT is not, it's a stop codon, then it is a nonsense mutation. If the ZAT is it's a codon for another amino acid, it is a missense mutation, okay. Okay, so in this case, let this be a missense mutation, which means ZAT is coding for another amino acid. Then what we will do? We will make a tRNA which is complementary to this make a tRNA whose anticodon arm is having the codons complementary to the ZAT, and it carries the same amino acid, okay. So what will happen? That mutation can be suppressed. So this is how we can correct this frame shift or nonsense or missense mutation using genetically engineered tRNA. So this is something that we are introducing, is genetically engineered. But what I have shown before this is normally that happens. So this principle that we are using in what is called as the therapeutic tRNAs, okay. So that is called therapeutic tRNAs. Now finally going to another question, like neonate blood sample taken, which test will be done for detecting the metabolite level for inborn errors of metabolism? So definitely we should know that for a metabolite, so question here is metabolite. So for a metabolite, it should be something which can detect a chemical compound. It's a biochemical compound that is called a metabolite. So definitely it should not be next generation sequencing, because this next generation sequencing is for DNA, not for the metabolite. Urine analysis, like it's a vague term. We cannot give that as answer. ELISA can be a screening method, but usually as a screening method we don't. Diagnostic we use the ELISA. For diagnostic purpose we use the ELISA. Which in detecting a metabolite, like neonate sample is taken, is usually for a screening test. Screening test we can use tandem mass spectrometry, and ELISA, if it was there, I'm not sure whether ELISA was there in the option. If ELISA was there, that can also detect metabolite. I'm pretty sure, like we know that it can also detect a metabolite, especially ELISA is that metabolite should be a protein. ELISA is a technique where antigen and antibody can be detected. So it is concerned with the protein metabolites only, not with all metabolites. So this question is not saying that a particular protein metabolite like that. It is generally saying, or just saying that it is a metabolite. So saying a metabolite says that it is not a next generation sequencing. Urine analysis, it is partially correct, but it is not a completely correct answer as long as tandem mass spectrometry is there, right? The tandem mass spectrometry is considered as the gold standard investigation of any metabolite, any metabolic disorder. Any metabolic disorder, the gold standard investigation is tandem mass spectrometry. So if it is there and metabolite is there, then definitely the answer is tandem mass spectrometry. And one more thing I'll ask you, tandem mass spectrometry detect the metabolite using based on which principle, or based on what the metabolites are being separated? It is based on mass to charge ratio. So based on the mass to charge ratio, the metabolites are getting separated. Each and every metabolite is having a unique mass to charge ratio. So we know which metabolite is having that particular mass to charge ratio. So from that we can find out what is the metabolite excreted in urine or what is the metabolite present in body fluids. We can find out. So here the answer is tandem mass spectrometry. So we have finished the questions of INI-CET May 2024. I just want to conclude with this quote. Everything you want is sitting on the other side of fear. So boldly, you just see the other side of fear. You should be able to see that. Then only success can be yours. All the best for you. Thank you.